Hello everyone, in this video we are going to discuss about apoptosis. Apoptosis is a programmed cell death. It takes place in two pathways, that is intrinsic pathway and extrinsic pathway. First, coming to the intrinsic pathway, it is also called as mitochondrial pathway. Whenever there is any stress due to medication or any radiation exposure or any fever, they lead to activation of the apoptotic factors. These are BAT, BID, PUMA, NOXA and BAX. So these inhibit the anti-apoptotic factors like BCL2, BCLXL and MCI1. These anti-apoptotic factors regulate the mitochondrial outer membrane. So whenever there is inhibition of these anti-apoptotic factors, there will be formation of pores within the outer part of the mitochondrial membrane. So there will be mitochondrial permeability increases. So this is mitochondrial permeability transition. This leads to release of the cytochrome C. These are the cytochrome C which are released from the mitochondria, and they, along with the apoptotic protease activating factor 1, and these also form the procaspase 9 cluster. All three together leads to formation of apoptosome. So this is an ATP dependent thing, so apoptosis requires ATP. This apoptosome will lead to caspase 9 activation, and which further leads to caspase 3 and 7 activation. These are the executional caspases which leads to formation of apoptotic bodies. Second is the extrinsic pathway. This is a death receptor pathway. Here the death ligand and a fast receptor binds first, and they cause the recruitment of the adapter proteins. The adapter proteins are TRAD and FAD. So along with this adapter proteins, procaspase 8 will cause formation of death inducing signaling complex, also called as DISC, and this activates the caspase 8 and in turn activates the executioner caspases like caspase 3 and 7, and finally leading to formation of apoptotic bodies. So we can remember like this one. First, intrinsic pathway can be remembered with the mnemonic of BAM-CAC, that is first BAD inhibits the BCL2, that is anti-apoptotic factor, and it further activates the apoptosis and leading to increase in the mitochondrial permeability, causing cytochrome C release and apoptosome formation and cascade activation, caspase cascade activation. So let us see the differences between the apoptosis and necrosis. Apoptosis is a programmed regulated cell death. Necrosis is an uncontrolled accidental cell death. The triggers are DNA damage, any withdrawal of growth factors and immune signals. Necrosis is due to ischemia, toxins, infection and trauma. Apoptosis is ATP dependent, necrosis is not ATP dependent. This is important. Cell size will shrink in apoptosis, cell swells in necrosis. Membrane is intact but formation of blebs occur in apoptosis. Membrane is disrupted in necrosis. In apoptosis, membrane is intact. In necrosis, membrane is disrupted. Inflammation is absent in apoptosis, it is present in necrosis. DNA fragmentation occurs orderly in apoptosis, random in necrosis. Nuclear condensation and fragmentation, that is karyorrhexis, occurs in apoptosis. In necrosis, there will be pycnosis, karyorrhexis and karyolysis. This condensation along with fragmentation and dissolution of the nuclear material occurs in necrosis. Enzymes involved are caspases, that is initiator and executioner. Executioner are caspase 3 and 7, as we already saw. Caspase 3, 7 are executioner caspases. In necrosis, lysosomal enzymes are involved. Here apoptotic bodies are formed, and in necrosis, cell rupture along with spillage of contents which leads to inflammation. Apoptosis can be both physiological and pathological. Necrosis is always pathological. So coming to this is a diagram showing apoptosis versus necrosis. In apoptotic pathway, first there will be cell shrinkage with condensation of the chromatin. There will be loss of water. This leads to formation of the blebs. Cell blebs are formed and apoptotic bodies are formed. Then these apoptotic bodies are phagocytosed. Next, in necrosis, there will be cell swelling. Water will be inside. Water will be drawn into the cell, and the membrane will not be intact as seen in the apoptotic pathway. Here we can see membrane is intact, but it is being fragmented. That's it. Here the membrane is actually not intact. So This leads to formation of necrotic bodies, finally cell lysis and release of the intracellular components, which causes inflammation.